I was listening, so that was great. So thanks so much for the invitation. I think that this is actually the first time we're in this meeting, and I actually have 4 of our A team here with me, which I think you all maybe already have met and seen some of your posters. Nestor actually is taking over our molecular biology and genetic engineering unit, so he's the one here who should be learning everything about how to work with new novel yeast expression systems. Zujun and Yunsun actually runs our process development unit, both upstream and downstream. So anything that requires scalability and optimization of processes, they're here to learn and share. And then Wen actually is our director of our analytical and development unit, as well as quality control. And I have to say, he's the one who is our rock star SARS scientist. He started it all for us around 10 plus years ago now. And that really enabled us to then be ready for COVID-19. But I'm going to actually tell you a story about how we love Pichia, and that's why we're here, and how we actually think it's a very nice technology that can be used to advance a lot of global health vaccine type of projects. So next slide. A couple of disclosures, which you can quickly read. Next slide. So our group have more than 24 years really specializing on a very unique group of diseases called the neglected tropical diseases. Maybe you've heard about some of them, maybe you've never heard of them. They are all sorts of parasites, protozoa, some can be, of course, viruses, some bacteria, but they have some very unique characteristics. They're definitely not neglected because of the amount of people that afflict. Clearly it's not a neglect because of that, because we have at least, what we call the bottom billion population of this world, which now is probably more like 1.5 billion people that are afflicted by one or even more of these diseases. But they're major cofactors. They actually go alongside many other diseases, including even non-communicable diseases, and especially, of course, the big 3, right? The malaria, the HIV, the tuberculosis plus. They're pretty huge cofactors in non-communicable areas like blood pressure or cardiac health, nutrition, and many others. In addition, unfortunately, because they primarily afflict populations who generally also don't have a lot of healthcare access or even education or even sanitation, they are very linked to poverty, and therefore generally those populations which now we have so many social forces like conflicts or even migration or climate change, right? They're usually also, very much having a big role in climate science. And they're not killer diseases for the most part. They're actually morbidity diseases. And that's actually a pretty hard sell, believe it or not. Even though we have a lot of people afflicted, the fact that they're not killing people, it's hard to explain to people why therefore they would be important, even though you can quantify the number of years that a person can lose because they live in a constant disability. That's the disability adjusted life years. And for some reason, we humans, as much as we, of course, we want to avoid death, we seem to be very happy with people living in the world with very bad health. And I think that's why we think that addressing these diseases, especially through vaccine, could be something that could really restore and make people healthier and therefore more productive, right? And that's how we also now work very heavily with economists to try to make that case that it's not only a public health case, a health case, it's an economic case. And of course, we saw this clearly with COVID, right? That you disrupt somebody's health, you disrupt everything in the world, right? And we now know that this compendium of, diseases have huge losses of productivity, which of course then, you can quantify by economics. So next slide. So we actually, we started working on this area of tropical diseases, again, more than 24 years ago, and we started in Washington, D.C. at the George Washington University. And I'm a co-science partner with Peter Hotez. Maybe you've, of course, heard of him. He's always usually in CNN and the media, and maybe from the European side of the crew, maybe not as much, but clearly he's a big advocate for not only tropical disease science and vaccine science. But now 13 years ago, we moved down to Houston. It was a very unique recruitment. It was a recruitment that was from an academic perspective, Baylor College of Medicine, which is a health sciences university, but linked with a recruitment with a hospital system, a pediatric hospital system, which is Texas Children's Hospital. And we found this very interesting and unique because if you actually look at the history of vaccine development, many, if not the majority of vaccines developed throughout many years, it's always a partnership between an academic hat and a children's hospital. And you would think that that makes sense because for the most part, even the traditional vaccination strategies are pediatrics, right? Now, of course, we have a lot more adult strategies. So we were very happy to come down to this enormous medical city. How many of you have been in Houston? Oh my God, you have to come, right? I mean, you've been there, yeah, I know. But you have to come. It's the footprint of downtown LA. There's more than 40, 50 institutions, all related to health, right? And as you know, everything is big in Texas, right? So, and for us, it was great because then our goals, audacious goals, we thought that we could have certainly, a good place to be, right? And there we set up this a little bit complicated but very exciting infrastructure, right? We were recruited to become a school, so we are like the fourth school of Baylor, medical school, graduate school, health professions, and then tropical medicine. And you may always ask, what is a school of tropical medicine? To be very honest, is whatever you guys want it to be, right? Because we don't have accreditations, we don't have any kind of like things that we really have to be scripted to the training and educational program. So we are whatever you want us to be. And we love that because we have a lot of flexibility in our educational activities. And then, like I said, we linked up with pediatrics. So we also run a division of pediatric tropical medicine. And within the division, we're around 20 faculty, so we're very small, very lean, small, but very passionate. We actually have this Center for Vaccine Development that I'll be talking to you about. Next slide. And we really do, to be very honest, a whole interdisciplinary type of work, right? Of course, with the mission of looking at tropical infectious emerging diseases, but we span the whole translational gap, right? Basic research all the way to trying to enable that translation from the bench to the clinic. And we have a whole array of programs for different types of diseases. In fact, our portfolio is quite large. But we also integrate this concept of health clinical care. So we actually have 2 tropical medicine clinics, one adult and one pediatric, where we see, believe it or not, people would always think that it may be migrant health, which we do, because of course tropical diseases come from migration and people from Latin America or Africa. But we also have a lot of transmission of many of these diseases in the Gulf Coast region and also in Texas. And of course, we have this education, Tropical Medicine Academy, we call it. And we do things with the premise of everything we do, we cannot be good at everything, so we do it in partnerships, and especially our vaccine center is anchored on what we call product development partnership model, so we partner. Most of our vaccines have some link with drug treatment, right? So you will usually parasites, you deworm, and then you integrate like an immune therapy or a vaccine treatment, scheme. But we also do the basics, right? Epidemiology, vector control, host pathogen relationships, immunology, so you name it. And of course, we train as we go. And since we do a lot in partnerships, we love training even at the global level. Next slide. But when we looked at how should we set up this infrastructure for advancing vaccines, and at some level, some diagnostics too, we learned from all these 24 years that there's a few lessons that we had to set up, including some that I'll tell you at the end of the talk. But we needed to select what we call the strategy, right? What we call the product development strategy. And talking to a lot of the mostly pharmaceutical, biotech, and those who actually do this for so-called for real, like the big pharma, there were various models that they were recommended to us, but one of the models is this so-called rapid entry into phase 1. Meaning, usually where things fail is when you get into the clinic, because it may fail in safety, or it may fail in, immunogenicity, or there's something. So rapidly you can get into the clinic, faster it is. So that required that when we had to prototype the vaccines that we wanted to bring to the clinic, we had to, of course, be robust, reproducible, of course, of high quality, make them under GMP, but that we should not really waste too much time in really finessing this production process, because we may be able to mature it later when we created more industrial or large-scale partnerships, but that the first thing that we really needed to do is to try to get them into the clinic, so that we didn't have to spend too much money and too much time, right, refining. And for that to happen, we then needed to, of course, have a very good antigen discovery pipeline, even though we usually never get money to do more than 1 or 2 candidates at a time, which of course is very different from the drug developers that they do like lots of leads, right? Hundreds of leads. We usually get like 1 shot on goal. And then that's how we decided that for us, we needed to try to limit the number of technologies that we would do these prototype vaccines, and that's where recombinant protein technologies were our selection. Now we, of course, work in others, but because we thought that they were easier, less concerns with the regulatory pathway, they were very well understood and known. And then we decided to set up our infrastructure. So what would we do versus what would our partners do? And this is what you see in the slide next, which, prior, I was gonna show yeah. So in our labs, as I said, we do the discovery sometimes, but when we don't, we obtain a target from a partner. But then we do the engineering into the prototype expression system. We do the feasibility of expression. Most of the times we do some of the preclinical animal models or even in vitro testing. But the most unique thing is that we indeed, do the scale up and process development. I think we have the ability of doing up to, if we wanted 20-liter scales, but we usually try to limit to 10-liter scales. And then the magic is, how do you then transfer that technology alongside all that criteria for release and stability? And then work with partners to do the toxicology evaluation, and then we actually hold our own regulatory dossiers. So I work with the FDA, with the ANVISAs, with the COFEPRISes, with whoever regulatory agency. And of course, then we partner with those who can do the clinical trial sites. So next slide. And that's why I'm not gonna tell you how we love yeast. It has a lot of advantages and a lot of challenges too, but at the same time, that's why we think that yeast is quite versatile, right? For us to, at least is our first choice to look at first, and then we decide whether we need to look at a different alternative platform. Next slide. But the real reason why we love the yeast is because there's a huge, strong, robust ecosystem of producers around the world, knowing that most likely we would never get the big Mercks, GSKs, or whoever to join us. We would have to find others who could really adopt this yeast technology so that they could make it into a real product, right? And there's an ecosystem of that. In fact, they are actually called the Developing Country Vaccine Manufacturing Networks, and they span from Bangladesh to China, India, Indonesia, some in Latin America. And of course, the reason why they know how to work well with yeast is because most of them also produce their own hepatitis B vaccine. And as much as it's not perfectly exactly the same as how we use the yeast system for monomeric proteins, versus particle proteins, they definitely have the knowledge and the infrastructure. And for the most part, the way that these vaccine manufacturers work is that they work for the public good, right? They usually have, yes, products that are of commercial interest, but most of the times these vaccines are made at very large scales, very, very, very cheap. And then they negotiate with countries or through Gavi or through UNICEF or the Pan American Health Revolving Fund, and they pretty much distribute, for the most part, around 60% of all world's vaccines, right? So we had an option. Next slide. And so this is how we envision our model would look like, right? So very simplistically, you have to go, of course, through all the discovery, preclinical, phase 1, phase 2, phase 3. Then that's what we call the translational gap, the first valley of death that you may hear all of a sudden. That's that first in human model that we wanted to be in. But there had to be eventually a point where we bring in these DCVMNs, where they take on the manufacturing maturity while also taking on post-phase 2, because usually when we work with pilot producers, that material cannot go for industrial, right, or for human licensure. So we can do small lots. Usually we work with Walter Reed, for example. The US Army has a pilot facility that we can do for small lots, but those pilot facility lots cannot go for licensure. So eventually we would always need to find someone to take it over. And they would have the large, and very difficult process of not only doing the large manufacturing, the phase 3/ even post-introduction phase 4s, they would have to, unfortunately, they have a different tier of WHO review and pre-qualification, and then they would have to figure out how to do implementation studies and eventually work with the financing procurement systems and eventually convince the governments that these vaccines have to be integrated into the vaccine schemes, right? And who would buy it, right? So, but that, albeit, was our audacious goal, right? Our dream. Next slide. So as we go along, we said, okay, what's gonna be in our portfolio? Our portfolio actually started, again, 25 years plus ago with, targeting hookworm infection. I'll give you a little bit about that program, where we're at. We also started a schistosomiasis program, a Chagas disease program. I think Wen had a poster on our work on leash. We also have a very exciting Lyme disease program. We partner with a few groups working on onchocerciasis. But for the most part, we slowly managed to go through this translational activity, moving these prototypes from the bench to the clinic, to the point where actually, in fact, our schisto and hookworm are in the phase 2 clinical trial stages. Chagas is entering phase 1 this year. And we're excited about that, albeit we don't really have that industrial model yet. Next slide. But something happened when we sort of actually were moving down to Houston back in 2010, 2011, where, of course, there was a lot of changes, shifts into what kind of interests are around the world, pandemic preparedness, biodefense. And we ended up, the group actually that works on onchocerciasis with us, they also had an interest in coronaviruses. There was a big group working on coronaviruses. And when we moved to Texas, Texas has one of the BSL-4 labs, like the big national laboratory labs, and a lot of scientists in the UTMB, University of Texas Medical Branch, actually work on these very nasty viruses. And they called us and they said, look, we have this RBD, and we don't really know how to fashion it into a protein vaccine, critical path, regulatory enabling. There was one call back from the NIH. We said, sure, let's apply. And we actually got a grant from the NIH, probably the last grant that they were giving at the time for coronavirus research. And we actually, in fact, that's when we recruited Wen, and Wen fashioned this SARS RBD We actually went all the way to making a clinical lot, and then we went back to NIH and BARDA and all the people to be, and we said, okay, we now have this vaccine prototype. What do you guys want us to do? And they said, that's not a priority anymore. So we not killed the program. We kind of continue working under the table, learning a little bit about the stability, the product characteristics of this RBD made in this yeast platform. And then, of course, it's history, right? Then they actually called us and said, now MERS was important. Can we look at MERS? So we shifted a little bit with MERS. We actually never could make MERS in yeast. We actually ended up testing it in mammalian cells. But then, of course, we were very rapid to pivot to COVID-19 because it was very similar strategy. So next slide. And so that's what we did, right? We engineered this RBD, which now we have all sorts of variants, RBD seed stocks, including, of course, the recent XBB 1.5 or 1. Whatever it is number now, or JB, whatever it is. So we create the engineered seed stocks. We, of course, full characterize them. Then we do some process development. Some of them are much more advanced. Some of them are just basic, without much optimization. Of course, we now have all the assays for their stability, indicating, and even release. And we started shopping it around, right? At that time, we started shopping it around, to see who could adopt this technology. While we were also doing some partnerships to evaluate formulations and evaluations. So next slide. So the way that it actually worked, we didn't have a lot of people lining up to come and say, give it to us, right? But we were lucky to have a few. I'm just highlighting a couple, which were, of course, the success, where we showed that that model that we wanted to use actually did work, albeit for a pandemic, preparedness situation, right? So we actually started with doing evaluative material transfer agreements, just sending them our research stock, sending them our data, see whether they could do their due diligence. Eventually, we did do a tech transfer agreement, and because we don't have any patents, at least for our engineering or our molecule, they were able to do non-exclusive licensing. Of course, both Biological E and Biopharma in Indonesia, so this is in India and this is in Indonesia, they were very savvy with using yeast-expressed products. Corbivax, which is the India product, just recently, believe it or not, 3 years later, got the WHO emergency use listing, which is good, the pre-qualification. It was evaluated in India against Covishield, which is the AstraZeneca version made in Serum Institute of India in India. But then the Biopharma was amazing because we also started working in the Middle East area a few years ago, and we were always very culturally intelligent to assess what kind of things you would need for Muslim majority countries. And in fact, when we created our engineered research stock for SARS-CoV-2, we worked with GenScript to make sure they gave us all the records to show that there was absolutely no animal-derived material. And when we actually got audited by the BUMRA, which is the regulatory agency of Indonesia that came with their religious, unit to review, we were able to support their documentation, and eventually the Indovac vaccine was actually certified as a halal-ready vaccine. So that was, again, those little thingies are like, precious, right? Precious moments. So we're very happy. And I have to say, and I'll show you again in my lessons at the end, it didn't take them that long. And I think it's because they were so savvy with yeast systems, right? Like BioE, for the initial, they took around 17 months before they got their initial EUA. So it wasn't that far behind of RNA technology. I have to say probably that they could have gone faster if they had more money and if they had more support, and I'll show you why. Our recombinant proteins, they really didn't get a lot of global support. Now that we transferred to BioE, the XBB version, it took them only 7 months, really, pretty much. Right now, they're all waiting for their final authorization in India. So as you can see, it's a pretty agile system if you really want to do this, right? Next slide. So our story, you can scan the QR code. We have a very nice expert review vaccines kind of review that kind of tells you the whole story, but very proud, right? I mean, we could not have done it without having, all the teams helping us really transfer our technology. And I have to tell you, this was very interesting because it was not just, here's the seed stock, here's our records, thank you, goodbye. They were really engaged with us. So we learned an amazing amount of information of how do you go from 10 liters to thousands of liters? How do you go by navigating this complexity of developing a real product, right? I was just in India last week, and it was just lovely to finally see in person where they were producing, both Corbevax. We still have to go to Indonesia, but 100 million doses have been administered. So I think this is our gift to all of you, I guess, to all the world. So we're very proud. Next slide. But now I want to shift back to tell you, so what is this going to do for us now and how to try to advance our other portfolio, right? Because it's not the same as having a pandemic. We don't have emergency. The people are not dying of this thing. There's no money. There's never any money. There's no real, interest here. But it's a very interesting program. In fact, these are animals, right? This is a hookworm, so it's a big worm. It's like 1 centimeter. It hooks into the mucosa of the intestine, but they're avid blood feeders. And in fact, 20 plus years ago, working with a group that now is in Australia, we started looking at the digestive system of the worm. How do they digest the blood meal, which is their food, right? And we uncovered a whole pathway that, of course, utilizes multiple enzymes, aspartic enzymes, metalloenzymes, but we also found a molecule that detoxifies the heme, right? So kind of make the worm go to the bathroom. So we selected actually a couple of candidates, the glutathione transferase and the aspartic protease. The aspartic protease, I'm not going to talk because we could not make it in yeast. We ended up trying a tobacco plant technology, which was a bust. But the GST was made in Pichia, and it's amazing how well it's made and how robust it's made. So next slide. So it is a heme binding and a detoxification. We have a lot of data on it, not only of the molecule itself, but also preclinical. It is also formulated on aluminum. That's kind of our base. Usually we do proteins on alum, and then we play around with immunostimulants. In this case, we also played with a TLR4 agonist and a TLR9, which is the CpG. Next slide. And we have, of course, all the data is published, right? All the cloning, engineering. So if you're interested in hookworm, GST, you have plenty to read. Next slide. But then the question was, how do we move this in the clinic? And the strategy we use here is we always try to go through a US FDA, because I think it's good practice, even though we don't maybe ever need to use the product in the US, even though we did find hookworm in Alabama recently in the sewage, in very poor areas where they have very poor sewage systems. But the strategy is that we started doing first-in-human studies, again, rapid into first-in-human in non-endemic areas, US, and then we moved to endemic areas, Brazil, Gabon, and USA. Netherlands is because we had a partnership through one of these European Commission framework package projects, so they helped us a lot. And with the group that actually we left behind in GW, which are our clinicians, they did a variety of phase 1 studies, but then the question was, how are we going to really show efficacy? And if you want to do a real field efficacy study, one, it requires a lot of people, a lot of money, and a lot of time, because you're going to deworm, most likely, vaccinate the people, send them out to their daily lives, and then wait to see that they get reinfected. But you don't have a control. When do they get reinfected? How much will they get reinfected? So it's just, very difficult to put a lot of money without not really knowing whether this thing is gonna work. So one of the things that they were suggested to us, which is now pretty much a little bit in fashion, is to develop a controlled human infection model in the lab, right? Or in the clinic, meaning that we take a person. In fact, we have donors, people who actively have hookworm infection, and they give us their feces, and we extract their larvae out of those feces, and we make it into a GMP production of larvae, right? And then we bring another cohort where we vaccinate, and then we challenge them. And in fact, we did that under, of course, US FDA IND. And then the NIH gave us money to indeed do that, do a 3-dose scheme of vaccination with a cohort. And then we challenge, with the larvae. And in the next slide, we were outstanded by the results. We just got, these are hot off the press. So in the left side, the way that you measure if there is, in fact, infection is by looking at the number of eggs that you find in the feces, because we cannot, of course, count the worms in the intestine. But it's a reflection of the larvae that became adults in the intestine, the female larvae mating with the male, and then being fecund, laying eggs, and then you measure the eggs in the feces. So we were very worried because we were not sure if this was going to work, right? In such a small cohort of people. We tried the vaccine with no vaccination, just challenge. We tried with the vaccine on aluminum and the TLR4 agonist. We tried just the vaccine on aluminum, but look at what we got when we put the vaccine on aluminum and CpG. And that coincided not only with much higher antibodies induced by the vaccination, but an amazing recall of the immune response when you were challenged with the larvae. This also doesn't show here. We also evaluate eosinophilia, because that's also a reflection of how control is the infection by not producing eosinophilia or IgE. And we also found that this group, which is practically 100% protection, it had very low eosinophilia. So we're excited about this. I hope you are too. But the question is, what do we do now, right? So next slide. We are ready, according to the technology readiness, that ideally we would like to now go back and do what I told you, right? Refine the process, do industrial levels, find that partner that would, drink the Kool-Aid, that this is worth advancing, and then maybe going into, advancing, maybe repeating this challenge study, maybe going and doing an actual field study. But the question is, how are we going to really convince? How would you even bring in a hookworm vaccine into the real scenario, right? One idea is maybe combine it with a malaria vaccine, maybe doing co-formulations with other types of vaccines. So we are still trying to figure this out, but at least we're very excited that maybe we can, now that we have the end of one of our COVID vaccine with some of these partners, maybe they get excited about this. Next slide. So I'm going to just finish very rapidly with the lessons that we learned, right? So why do I think and why we think as a group that we were successful? Scientifically, that's what we were trained for, right? So as much as science has a lot of technological challenges, we are scientists. But I think we're not prepared of understanding regulatory science. And I think that last 20 years, if I say that where we have done a great investment with our teams, is to really learn the regulatory science. It's not the same to develop a product for a manuscript, to develop a product and put all the documentation for a dossier for a regulatory agency. The way that you do document control and quality control and a quality assurance was prime, right? That's why BioE and Biopharma loved us, because we gave them documents that they just immediately stuck into their dossiers. They didn't have to redo any of the work. The second lesson is that, albeit all this, that science is great and that regulatory, you can learn it, we are humans and we have to deal with social issues, right? Acceptance, hesitance, understanding who will use it, the cost, the finances. So I don't think we have figured this one out very much, but we have to be aware. So in fact, right now what we're doing back about hookworm is this whole value proposition, right? How can we really put in a pitch presentation that really will convince the funders? The third is that it's complicated, right? These are vaccines that definitely are for public good. There's no market, right? Or certainly failed market. But there is an opportunity of reevaluating how we position the technology, the open science, the funding, the linking of funding. And then the last one, which I'm going to leave there, next slide, please. It's the fact that where I think was very difficult, especially during COVID, is this concept of the failures of policy and the failures of politics. That's vaccine diplomacy. And how many of you have seen the show Slow Horses? I love it. I feel that we were in exile and we were always considered like the screwed ups because we were working on yeast, which was not innovative, not a shiny new toy like, the RNAs. We were the matchbox cars. And as much as, yeast is a workhorse, we were really relegated, right? We were the ones who, and as academics, we automatically did not know what we were doing, right? And we had not the chops to work with the big pharma. So we need to change that. So I leave you with that provocative, thought is we are all the ones who can advocate for yeast. And we can advocate for yeast for many things. I mean, all the posters you were showing are amazing, right? From making milk to using it as a source of energy, but we need to tell people that this is the workhorse and that, we should use the yeast more and that it's not just, an old-fashioned technology that, has no innovation. You guys are creating that innovation. So thank you, and the next slide just shows you a little bit the faces of the group that worked with us during the COVID-19. So I appreciate it, and if you have any questions.
I'm sure there's a couple of questions. We have some time to do.
Thank you so much for your nice talk. I think vaccines are probably the most important invention, right, for a scientist to save the world at the end of the day. So regarding Pichia, I think you restored my faith in that, Pichia is indeed can play a role in that whole aspect of vaccine development, and there's plenty to do. But going to your hookworm example, it seems that there is a revaccination needed. Is that correct, based on the data you showed?
Say that again, is an infection what?
For the hookworm vaccine, is revaccination needed for that?
Yes, so good question, because that's usually what we always get, right? Why do you need vaccines if you have drugs? Why do you need vaccines if you can, of course, educate people to go to the bathroom correctly or don't defecate in the fields? Because, of course, it's a human transmission. But it's been decades and decades and decades of deworming programs that are just not working, right? Because, of course, there you have the thankfulness that the pharmaceutical companies donate the medicines and they're cheap, but that's a burden to the countries that they have to do this deworming all the time, right? Some of them, they are school-age specific, some of them maybe even community-based, but without, where do you see that a single intervention usually works, right? You certainly don't use that in malaria, right? You still use bed nets and you still use other things in addition to the new vaccine. And the challenge is that we're already started, certainly in veterinary field, there's a lot of resistance. We haven't maybe documented as well in humans, but we do have some idea that there already is circulating resistance when you use albendazole, mebendazole in humans. So clearly we need a complementary, a companion technology to go with.
So thank you. The other question I have is regarding, so you focus really on the human market. How about the animal part of this?
Yes, so in fact, we started with a veterinary approach. So all these genes were originally cloned from the caninum, so the dog hookworm, and even selenicum, which is the hamster or the rat, mice hookworms. So all these molecules are very well conserved, and in fact, we even think that the Ancylostomos caninum would be a great veterinary vaccine. But there's, again, not a lot of market for that. But you're right. I mean, we could.
The pet market is really big, right? So
You know,
it
back-
could be a way in.
Yeah, back in the 70s, there actually was a licensed hookworm vaccine that was based on inactivated or attenuated larvae for dogs, but it was very complex to produce and maintain, and people were not very happy to see their dogs, in fact, being vaccinated with larvae. But you're right. I mean, we always have in the back of our minds, how can we do dual use, One Health use. But again, it's we of course want to prioritize human health, but then if you can also kind of add on the veterinary field would be great. We do that for all our vaccines. Chagas is a veterinary issue. Of course, schisto maybe less, but still, right? We can look at some of that, and especially we also have vector control involved. So yes, good points.
Thank you so much.